Hello everyone, welcome to lecture 6 of our short course, and in today's video we will dive into the concept of heritability. We will explore how heritability is studied, starting from early methods and moving to modern approaches, as well as discussing the challenges in this fascinating field. So let's dive in. The concept of heritability arises from a simple yet challenging question. What causes variation in traits? There are two primary components contributing to trait variation. One, genetics, for example, DNA differences between individuals, and two is environmental effects, for example, differences in nutrition or lifestyle among individuals. This interplay between genetics and the environment is often referred to as nature versus nurture in the literature, and it gave rise to the concept of heritability as we understand it today. Heritability is simply a metric that quantifies the proportion of trait variation attributable to genetic factors. Its values range between 0 and 1, where a heritability value of 0 indicates trait variation is entirely due to environmental factors, and a trait value of 1 indicates that trait variation is entirely due to genetic differences. Let's explore the initial methods used to measure the heritability of traits. Historically, these methods were primarily based on studies involving twins. As shown in the model on the left, commonly referred to as the ACE model in the literature, heritability can be estimated by analyzing the genetic correlation between monozygotic or identical twins and dizygotic or fraternal twins. Here the components of the model are additive genetic variance, which is the heritability whenever the trait is normalized, shared environmental variance, which we assume is the same between twins, and the unique environmental variance specific to the individuals. Those components are assumed to be orthogonal, meaning that they are statistically independent and do not overlap in their contribution to the trait variance. The model is outlined in the figure in the middle. The correlation between shared environmental variance is assumed to be 1, and the correlation of additive genetic variance are assumed to be 1 and 0.5 respectively. Since monozygotic twins share all their genetic material, whereas dizygotic twins share only about half of their DNAs. The core idea of this approach is that any additional similarity in trait variation observed between monozygotic twins compared to dizygotic twins must be due to genetic effects. This is because again, monozygotic twins share their genome completely, whereas dizygotic twins share only about half percent of their genome. And on the right, I outlined the basic calculations for estimating heritability using twin studies. Assuming that the trait is normalized with a variance of 1, then VA represents the heritability estimate and can be calculated directly using the formula here shown. And in the next slide, I will briefly explain the problems of heritability estimations under this study design. Now, let's discuss some key issues in the design of heritability studies in twins. Now, first, we had the assumption of equal environment. We assume that identical and fraternal twins experience same or nearly same environments, which may not always be true. The second issue is shared environment influence. The ACE model assumes orthogonality, that is, the shared environmental effects and additive genetic effects are independent and do not overlap in their contribution to trait variance. However, this assumption is not necessarily true in practice, and especially under scenarios such as gene by environment interactions. The third issue is generalizability. Results from twin studies may not generalize well to broader populations because twin studies are generally not drawn through random sampling. Instead, they often come from twin registries or specific recruitment efforts, which may introduce selection biases. And additional twins may share unique early life environments that differ from those of singletons. And these differences could limit the applicability of findings to non-twin populations, non-twin individuals. The last issue is the sample size limitations. Obviously, twin studies have smaller sample sizes. So due to these outline approaches, researchers have transitioned to modern genome-wide approaches. In the next few slides, I will be explaining modern approaches to estimating heritability. There are three methods I will be discussing, starting with the Heisman-Elston regression, or HE regression for short. And this method operates on a simple yet powerful argument. Related individuals tend to resemble one another in their phenotypes. The model is outlined on the left here. Yk represents the trait value for individual k. We assume that it is standardized. And pi kj is the genetic relatedness between individuals k and l. A is the intercept, and epsilon ij is the random noise. Under this model, heritability can be estimated. But you might wonder why we have this model, why heritability is here, and why we have this model. And in order to address this question, if you replace the heritability here with an m, which is the slope, and solve the model for m under some assumptions, then you would find that the slope is nothing but the heritability of the trait. And here I provided the detailed calculations. These steps are borrowed from Wischer et al. With minor changes and additional explanations added by me for clarity. And I will not go over through these, but if you are interested in mathematical details, feel free to pause the video and read through the derivations. Now let's now move on to the next method. The second method is restricted maximum likelihood methods, or RML methods for short, and the concept is straightforward. RML is used to estimate variance components in linear mixed model settings, or commonly commonly applied in GWAS. And on the left, I provided the LMM, or linear mixed model setup, which serves as the foundation for RML-based heritability estimations. And if you break down the RML estimations in steps, the first step is to remove or projecting out the fixed effects from the data. This adjustment ensures that only the variance components of the interest remain for further analysis. And after removing those, we use maximum likelihood approaches to estimate corresponding variance components here. And these components capture the genetic and environmental contributions to the trait variance. And then finally, heritability is calculated by plugging the estimated variance components into the heritability formula. And specifically, heritability is defined as the proportion of total variance attributable to genetic factors. Both RML methods and HE regression methods require access to genetic data, and now we will be moving on to a third method which do not require access to genetic data. The last method is based on GWAS summary statistics, and it is called LD score regression. This method is slightly more complex than the previous one, so I will explain its core logic in more detail. Let's start by recalling the LD problem in GWAS settings. In GWAS, a found association by a SNP may not be always direct. It could be instead an indirect association caused by the linkage disequilibrium structure between the SNP being tested and other SNPs in the genome. For instance, imagine we have an LD block consisting of two SNPs only. And let's say SNP1 has a direct association with the trait, an estimated GWAS effect size of beta 1 hat. And SNP2 is correlated with SNP1 through an LD value of R, through correlation with R. Now, even if SNP2 does not directly influence the trait, it can show an indirect association with the trait due to the correlation between SNP1. In this case, the indirect effect size for SNP2 would be beta 1 times R. And I have provided here the mathematical details on the right side, but again, feel free to pause the video to check it out. The important argument is that even if it is not affecting the trait, it would be indirectly found associated with the trait due to this correlation. And here I provided a simple example where the LD block of two SNPs, but of course in real world scenarios it is much more complex and involving different sizes of LD blocks and interactions. And LD score regression addresses these challenges. And in the next slide, I will dive into the details of how this method works. Now, the main argument of LD score regression is based on this causal model, which provides a framework for understanding how trait values are influenced by causal genetic variants. The model assumes that trait values can be represented by two causal variants, GI, with their corresponding true effect size estimates, as shown here. And under this model, it can be demonstrated that GWAS marginal effect size estimates can be expressed as follows, like this. In other words, each marginal effect size is the sum of indirect effect sizes of other genetic variants, along with an additional random noise term. In this context, the term within the sum represents the indirect effect of a single SNP, and R corresponds to correlation with SNP between two SNPs, and here lambda I is the true effect size of true effect size of SNP I, which is 0 if it has no causal effect on the trait, and non-zero if it is otherwise. So this is the indirect effect of SNP I on SNP J. Okay, and then we run this through all genome positions. So I changes between 1 to the total number of genotypes, and then this total sum of indirect effect sizes together with some additional random noise term is giving us the GWAS marginal effect size estimates. Now, to make use of GWAS summary statistics, LD score regression relies on several key assumptions. While some of these assumptions are quite strong, they are essential given considering the data limitations. This is only based on summary results. It's not using any raw data or any genetic data at all. And these results, these assumptions can be grouped into three categories. One is about a kind of orthogonality of causal effects, indicating that the causal effects are assumed to be uncorrelated with one another. And the second group is the mean of the indirect effect sizes is assumed to be 0. And the last group of assumptions are about residuals. The residual terms, the random noise, follow specific distribution properties to ensure model validity. And under these assumptions, it can be shown that the expected chi-square statistic for a GWAS can be represented in terms of heritability here, and some of known or computable variables. In other words, the expected chi-square value is given by this formula, 1 plus nh squared divided by m times this term Lj, which is referred to as LD score of genotype J, and this can be calculated or used by pre-calculated models. So I should also add that for simplicity, I have not included fixed effect components in these models because it's already a complicated model. But for comprehensive calculations, including these fixed effect corrections, which corrects for the confounders, you can check the cited article here for details. But in summary, LD score regression offers a powerful way to estimate heritability using only GWAS summary statistics. That's the only thing you need to know at this point. With that, we have discussed the different methods to estimate heritability. And in the next slide, I will explain how to choose the right model for heritability estimations. We now have three key methods to estimate heritability, and each with its own variations depending on computational constraints or simplifications. So when I say HG regression model, it's not a unique model. There are different variations, similarly for ML. So there is no single correct method, but the choice of approach in literature generally follows this logic. Do you have access to raw data? If not, well, you don't have much option. The only approach is the LD score regression. And if you have access to raw data, and if you have sufficient enough computational power, then you apply RML-based methods, which are usually more accurate and robust, but computationally extensive. And if you have access to raw data, but lack sufficient computational power, or you are working with a large data set, then you use HE regression-based methods, which are simpler and computationally efficient. Each method has its strengths and weaknesses, and the choice ultimately depends on the availability of data, computational resources, and your study design. In the next couple of slides, I will focus on the challenges in heritability studies. The first challenge in heritability studies is how to define heritability for diseases, which are binary traits. For example, 0 for unaffected and 1 for affected. And unlike continuous traits, diseases present significant constraints because their phenotype values are limited to zeros and ones. To address this, we use two definitions of heritability for diseases. One is observed heritability. This is the heritability directly computed using RML-based methods under a linear mixed model setting. It estimates the heritability in the observed phenotype data, zeros and ones. And two is heritability under the liability model. And this approach assumes an underlying continuous liability for the trait. In this model, there is a continuous underlying trait where individuals are affected if their liability exceeds some threshold and unaffected otherwise. And this liability model follows a genetic additive assumption, here represented by this formula, YL equals GL plus EL. Under this framework, we assume that the observed genotype values can be expressed as a linear function of genotype values under the liability model. And using this assumption, we can compute heritability under the liability model as given in here. The only important point is that the slope is this linear function can be shown to be a value called Z, which is the height of the threshold that separate the affected and unaffected individuals. This approach adds flexibility and allows us to account for variation in the phenotype despite its binary nature. So we have two types of heritability in short for heritability of diseases. The second challenge is the missing heritability issue. In earlier studies, it was observed that heritability estimates derived from top GWAS hits were significantly lower compared to those from pedigree-based studies. For instance, for height, initially estimates heritability for GWAS was 0.1, while the pedigree studies estimated it at 0.8, a huge gap. And in 2010, Young et al. Conducted a groundbreaking study that showed common SNPs explain a substantial proportion of the missing heritability for height. They demonstrated that when considering all SNPs rather than only top SNPs across the genome, the heritability estimates could increase to almost 0.5, which is a significant improvement from the earlier estimate of 0.1. And importantly, this pattern was observed for other traits as well. In the middle of slide, I present a table taken from Wischer et al. In 2012, which compare heritability estimates from pedigree studies, GWAS studies that include only top SNPs, top hits, and GWAS studies considering all SNPs. The table highlights the discrepancy between pedigree-based estimates and GWAS estimates. And it also shows that there is still a notable gap remains between the pedigree estimates and GWAS estimates that uses all SNPs. This remaining gap is referred to as still missing heritability. And in the recent years, the availability of large-scale whole genome sequencing dataset has shed light on this issue. These studies have shown that a significant proportion of the missing heritability can be attributed to rare variants. For example, Weinstein, Jane, and Zhang et al. Estimated heritability for height and found that it's approximately 0.7 using whole genome sequencing data, which is nearly matching the pedigree estimates of 0.7 and 0.8. However, for BMI, heritability estimates still fell short compared to pedigree-based estimates. So this still remains as an open problem in genetics. Most researchers now believe that the addition of rare variant contributions will bring GWAS-based heritability estimates match to pedigree-based estimates, and any remaining discrepancy, if still remains, may arise from the strong assumptions inherent in pedigree studies, which might be violated easily. So this brings us to the end of today's video. Thank you for joining me today, and in the next video we will be delving into another exciting topic, Mendelian randomization, or MR in short. We will cover the basics of MR, the core logic and the intuition behind it, as well as its application and key challenges. I hope you are excited about this fascinating topic, and I will see you in the next lecture. So bye!